Okay, we were previously talking about water being a polar molecule, and it has areas of partial charge. So this is something you should just memorize. The oxygen, let me see if I can get a pen going here. Oh, I have to be in this, let's go into this mode. All right, so the oxygen side of the molecule, the back end of the molecule, has partial negative charges, because remember, it kind of pulls the electrons toward it and kind of hoards them behind it, behind the oxygen. Maybe you can think of it that way. And then the hydrogen nucleus, both of those end up with a partial positive charge. So all water has this fundamental layout, and remember, the angling of the water molecule is what creates this. And so you have this back end is partial negative, sort of two areas of the front side, I guess you could call it, would be partial positive. And so when you have a compound or something that's charged, or even something that's partially charged, but we're going to use a fully charged example here, when you mix salt into water, it dissolves. And what does that mean? Well, it means that the chloride ion and the sodium ion get kind of separated from each other, because the water molecules, and this is shown in a two-dimensional picture, but you really need to think of it in 3 dimensions all the way around. The chloride ion all the way around is being touched by the partial positive hydrogen side of a whole bunch of water molecules. The sodium ion is being touched all the way around by the oxygen side, which is the partial negative side of these water molecules. It's just a natural attraction. The partial negative of the water is attracted towards the positive ion, which is the cation ion, if you recall. The negative ion attracts a layer of water molecules that surrounds it, but it's the partial positive side, the hydrogen of the water, that faces that. So when you look at this here, you can see it's directly being touched by the hydrogens. When you look at this one, it's being the sodium is being touched by the oxygens. And when you think about that concept, when you think about that in 3 dimensions, you can see that it's basically like a cloak. The water forms a layer, or what we call a hydration shell, or a sphere of hydration, but it just means a layer of water surrounding each of those ions, and it allows that salt to be then dissolved. So when something is dissolved in water, what it means is that it's broken apart, and each part of it is surrounded by a sphere of hydration, or a hydration shell. And that allows the item to be mixed into the water uniformly. All right? So if you have anything, let's say I have something and I don't tell you what it is. Here's a square, whatever that is. But I show the water molecules touching. This is a terrible drawing. But if I show the hydrogen end of the water molecule directly touching the object, that would indicate that the object itself has a negative charge. If I showed another item, here's a pentagon, whatever, and I showed the oxygen part of the water oriented towards the object, trying to draw water, then that would indicate that it would have to be, the object itself would have to be positively charged in order to cause that to happen. So you can actually look at the orientation of the water molecules, and that gives you a hint regarding the charge of the object that the water is surrounding. Okay, we're going to look at, because of the polarity of water and the number of hydrogen bonds in water, water has certain properties. Water has cohesion, water has adhesion, which is not on this list. It's on the next page, but anyway, and water has surface tension. So cohesion means something sticking to other things just like it. So water molecules sticking to each other. And that's because of all the hydrogen bonding. We talked about that already. Surface tension, it turns out that at the surface of any water, a drop of water, a glass of water, at the surface where it interfaces with the air, let's say, there is a little bit more, a tighter bonding of the water molecules just at the surface. And so you've probably seen a trick where somebody put a pin, like sewing a needle like a pin, and laid it, and you can do this if you want. You put a pin, if you're very careful, you can lay it on the surface of a glass of water and let go, and the pin will stay there, even though the pin is really too heavy. But it's because at the surface there is some strength, what we call surface tension, and it's strong enough to hold the pin. There are some insects that can walk on water because they're light enough that they don't break the surface tension. So the surface has more tension than you would expect, and it's because of a special structure of hydrogen bonding there. Now, adhesion, cohesion and adhesion. Adhesion is the tendency for water to stick to something else other than water. And so adhesion is why water will crawl up a capillary tube. A capillary tube is a very, very thin straw made of glass, and water will move up a capillary tube against the force of gravity because it actually sticks to and adheres to the side of the glass, and it pulls the other water molecules behind it. So the adhesion to the glass and the cohesion of the other water molecules that are being pulled behind it, and it will actually move up. And in a plant, you don't have glass, obviously, but you have very, very thin tubes, and they will pull the water up the tube, and those are the tubes that pull the water up the stem to the leaves. And so the capillary action is what moves water up the stem or the trunk of a tree. If you cut open any upright plant, you won't find like a big tube. It'll be hundreds or thousands of teeny, really thin tubes, because the water can move by capillary action only up a very, very thin tube. If it's too wide, it can't move up. But the capillary action becomes biologically really important for plants. Oh, wrong way. Okay, pH. This gets a little complicated. This does not have to do with hydrogen bonding, so put all of your hydrogen bonding thinking aside for a minute. Any solution has a characteristic we call pH. And you've probably heard of that before. And you've probably heard of the concept of acid or base. Acidity means how acidic, how much acid. Alkalinity or basicity, but we usually say alkalinity. Basicity would be the same, but it's not a word we use a lot. But alkalinity and acidity is a characteristic of any solution. Now, we talked about in biology, all solutions that you're going to talk about in this course and in almost every biology course are going to have water as the solvent. And any solution that has water as a solvent is called an aqueous solution. And that word aqueous means water is the solvent. A-Q-U-A-E-O-U-S. And you can see the word aqua in there, which in Latin means water. So aqueous, that's what this means. Aqueous. Liquid, aqueous. Okay, now, an aqueous solution is one in which water is the solvent. Now, the water molecule itself can break apart. Now, this is not something that happens very much. A teeny bit of water will break apart. So in any drop of water you get from your faucet, there will be a tiny proportion of the water molecules that will break apart, and they will form, what that causes is the formation of hydrogen ion and hydroxide ion. So that's equivalent to one broken up water molecule. In pure water, there are 1 times 10 to the minus 7 moles per liter, which is equivalent to 1 hydrogen ion for every 10 million intact water molecules. So it's very little. But that's the hydrogen ion concentration of pure water. The hydroxide ion concentration, I think you can tell, would be the same, because for every hydrogen ion that forms from 1 broken up water molecule, you'll have 1 hydroxide ion. So what we're saying is these are very, very tiny amounts in any amount of pure water. But you can add things to pure water. You can add just hydrogen ions. So if you add additional hydrogen ions, but you don't add any more hydroxide ions, then that water becomes acidic. Or if you add just hydroxide ions and don't add any more hydrogen ions, that solution becomes more basic or more alkaline. So the definition of acid and base: an acid has a higher concentration of hydrogen ions than pure water, and an alkaline solution has a higher concentration of hydroxide ions. All right, but I don't like to use that definition for base, so I'm going to give you an alternative. A solution that has, in biology, another definition of a basic solution is a solution with a lower concentration, lower hydrogen ion concentration than pure water. I like that definition better. It's more biologically relevant. So we end up with a definition in biology. It's not different, it's just a different way to look at it. A solution with a higher hydrogen ion concentration than water is called acidic. A solution with a lower hydrogen ion concentration than water is basic. So we won't focus on the hydroxide ions at all. All right. So we have the scale, and a scale is called the pH scale. So here it is. And the scale, it cut off here, but the scale goes to 14. It goes from 0 to 14. Here's 14 up here, and here's 0 here. And pure water has a pH of 7 right here. So that's like the dividing line. And any pH number less than 7 indicates acidic. So acidic is on this side of the line. Put acidic. And any pH above 7 is basic or alkaline. Put alkaline here. Okay. Now, the further away from 7 you are, the more acidic or the more alkaline it is. So when you go from 7, if you have a solution that has a pH of 7 and you compare it to a solution that has a pH of 6, the pH of 6 solution is more acid than the pH, has higher acidity or a higher hydrogen ion concentration than pure water. If you compare a solution of pH of 7 to one of pH of 8, it has a lower hydrogen ion concentration. Seawater has a lower hydrogen ion concentration than pure water. So acidic means more hydrogen ion concentration, and the more acidic it is, the more hydrogen ions it has compared to water. And alkaline is less hydrogen ion concentration. All right, so when you're in the middle, that's pH of 7. That's the standard hydrogen ion concentration for water. If the solution has significantly more hydrogen ions, then it becomes more acidic, which means the pH drops. So it's an inverse relationship. pH, if the solution has a high hydrogen ion concentration, then that would be equivalent to a low number on the pH. The higher the hydrogen ion concentration, the lower the pH number. And inversely, the lower the hydrogen ion concentration, the higher the pH number. And mathematically, there's a reason why that's true, but we're not going to get too deep into it. The other thing about this scale is it's what we call a base 10 scale. It's a logarithmic scale, log base 10. So you may be familiar with, there's a log 10 scale that we use for earthquakes. It's called the Richter scale. I don't know if I'm going to spell this right. Richter. The Richter scale is how we measure the intensity of an earthquake. So if you have an earthquake and it's a 4 on the Richter scale, and then there's another earthquake and it's a 5 on the Richter scale, the difference in intensity between the 4 and the 5 is not 1, it's 10 to the 1. So you have a 4 intensity and you have a 5 intensity. Those are actually related to the exponents of 10. So what's the difference between 10 to the 4th and 10 to the 5th? Not 1, but 10 to the 1. If you remember how you do your, it's a tenfold difference. So the earthquake that's a 4 and the earthquake that's a 5, the one that's a 5 is 10 times more severe or strong. So in a similar way, the pH is a log 10 scale. So if you have distilled water and you compare it to urine, one has a pH of 7, the other has a pH of 6. Urine has not 1 times more hydrogen ions, but 10 times, 10 to the 1 times more hydrogen ions than water. Black coffee, since water is a 7 and black coffee is a 5, black coffee has 10 to the 2 times more hydrogen ions than water. Tomato juice has 10 to the 3 times. So however many spaces you have to go when you're comparing to water, that number is an exponent of 10 in terms of how many times more acidic or how many times more basic something is than water. And being more acidic means having more hydrogen ions, and being more alkaline means having fewer hydrogen ions. So when you say, okay, what's the difference between the hydrogen ion concentration of water versus milk of magnesia? Water has a pH of 7, milk of magnesia has a pH of 10. So that's 3 spaces difference. But remember, that's not 3, it's 10 to the 3. So milk of magnesia has 10 to the 3 fewer hydrogen ions than water. So if you're going in the direction, this direction, it's fewer hydrogen ions. If you're going in this direction, it's greater hydrogen ions. All right, so that's how the pH system works. And you just need to know that it's a log 10 scale and be able to make some comparisons between solutions. If you know their pH, which one is more acidic. Of course, it's always the one with the lower pH number. And know that the pH is really an exponent of 10. And so you can figure out how many times more or less acidic something is than water. Okay, buffers. Really, buffers, we're going to talk about buffers just briefly. The most common buffer in the human body is called bicarbonate. It's literally the material in baking soda. Baking soda, the full name of baking soda is bicarbonate of soda, which is soda, sodium. I don't know how they ended up with this because it's not like it's not like soda like Coke. Bicarbonate of soda is sodium bicarbonate. And so it's really, your body makes it naturally from the carbon dioxide that you breathe out. But anyway, it's the most important buffer in your body. And it just prevents your body's pH from changing very much. So it maintains your body at a constant pH. And actually, the pH of your body is not 7. It's like 7.2 or something like that. So it's actually, your body's natural pH is a little bit, it's just slightly on the alkaline side, which is kind of interesting. All right, carbon. We're finally able to talk about carbon. We talked about oxygen. We talked about carbon a little bit. And you remember that it can make 4 covalent bonds. It wants to make 4 covalent bonds. And all life on Earth is based on carbon. Carbon is the backbone, so to speak, of all the molecules in living organisms. And molecules that have carbon are called organic molecules. So when you take organic chemistry, we're using the word organic to mean carbon-based. Now, I know that there is another way that you use the word organic, like you buy organic produce at the grocery store, that means they grew it without special pesticides. But this is a different, we're using the word organic to mean, not carbonic, carbon-based, sorry, carbon-based. So molecules that have carbon. The funny thing is, the only molecule that's not considered organic that contains carbon is carbon dioxide. Carbon dioxide is considered inorganic. And remember that carbon dioxide is also weird in that it's nonpolar, even though it has polar bonds. So carbon dioxide is a weird one. So the exception to organic molecules containing carbon is carbon dioxide, because carbon dioxide is considered an inorganic carbon compound. It's the only one that I can think of off the top of my head, inorganic. Again, I didn't make up the rules, I'm just telling you what they are. So that's the definition. Carbon dioxide is an inorganic carbon compound, but everything else that has carbon is considered an organic molecule. And so all the molecules in your body are based on carbon, and then they contain then, you know, lots of carbon and then some hydrogen, some oxygen, some nitrogen. So that's going to be the rule for all of the molecules that we look at. The simplest organic compounds are called hydrocarbons. And hydrocarbons, in this case, the hydro means hydrogen, not water this time, and then carbon means carbon. So hydrocarbons are molecules that only contain hydrogen and carbon. And the simplest one is called methane. Methane is carbon and 4 hydrogens. And this picture here, when you take organic chemistry, you draw molecules using this pie-shaped drawing and these dashes to try to indicate the 3-dimensional shape of a molecule. But we won't probably do that. We're not going to do that. We're just going to draw them flat. So we'll draw, like when we draw methane, we'll just pretend like it's flat, although it's not. You know, like this. But anyway, when you see something like this, they're trying to indicate a 3-dimensional nature of the molecule as best they can on paper. But there's really no limit to what hydrocarbons can be. Methane's the simplest one. And then you can have 2 carbons and 6 hydrogens. You could have 3 carbons and 8 hydrogens. I mean, it goes on, you know, pretty much indefinitely. But the carbon is sort of in the center of the molecule, and then the hydrogens are kind of surrounding. But anyway. Now, these are all covalent bonds. I know I told you before that the ... is something we use for the hydrogen bonds, but in this case, carbon to a hydrogen in the same molecule, and the ... gets bigger and bigger like a piece of pie, then that's supposed to be showing that hydrogen is behind, like this one over here. See how it goes behind? So that's a covalent bond that goes behind. We won't be drawing with that. We'll do it like this. All right. But when you take organic chemistry, you do a lot of drawings like this. Covalent bonds hold a lot of energy. Remember I told you it's really hard to break a covalent bond? But if you can break it, a lot of energy will be released from that bond. In fact, hydrocarbons are like fossil fuels. Fossil fuels like gasoline and oil, these are hydrocarbons, and so when you burn them, that breaks the breaks the covalent bonds, and that releases energy in the form of heat, and actually it moves the pistons in your car, and so that's an example. When you burn hydrocarbons, what you're really doing is breaking the covalent bonds, and that releases energy to move your car or something like that. All right. Now, once you have a concept of hydrocarbon, like I said, you can have 2 carbons. You can have 3 carbons, etc. And then you remember that each carbon wants to make 4 bonds. So if the backbone of a hydrocarbon looks like this, then you just make sure that each carbon has 4 bonds coming off of it. So this one does. And then you fill in hydrogen everywhere that you need to. I don't know why my mouse does that sometimes. It's messy. So like could be like this. There's really no limit. But remember, it's really not flat. We're going to build some models. You're going to get a model kit. We're going to build some models. And you'll be able to see that the molecules have certain angles. But when we draw them, we'll just draw them flat. Okay? And so you can have these long chains of carbon and hydrogen. All right? But then there's also a possibility of having a branched hydrocarbon. And so a branched one, they've kind of, this is from your book, they kind of highlighted in blue the carbon backbones. And then like this would be like a very simple example of a branched one. All right, and there's no limit to how many branches there could be. This is a ring structure. And then as long as each carbon has 4 bonds and each hydrogen only has 1 bond, then it follows the rules. It's fine. There's a double bond here, carbon to carbon double bond. And then you fill in with the extra hydrogens. So those are very, you don't have to memorize these, like don't memorize the names of them or anything like that. Don't memorize the structures. But if I showed you a structure and I said, you know, is it correctly, am I showing the structure, you know, correctly? You just need to know that each carbon makes 4 bonds, each hydrogen makes 1, and then if there was any oxygens, those make 2, and any nitrogens would need to make 3. So I could give you a structure and you could just check and verify that it's the correct one. Okay, so when you have molecules, you can actually have molecules that have the same components, like 4 carbons and that would be 10 hydrogens. So for example, we would write that C4, 4 carbons and 10 hydrogens, H10. But that just tells you the makeup of the molecule. And when you write C4H10, if we had our model kits, you could build with those components, you could build this molecule, or you could build this molecule. In that case, that's about it. But both of them are C4H10. And so these are actually different molecules because they have different shapes. Shape is very important in chemistry and biology. So you can tell the shape's a little bit different. So molecules that have the same formula, this is the formula, the makeup, but have a different shape, it's really about the shape, or the details of the structure, those are called isomers. Now we talked about something called isotopes earlier. Isotopes are when you have different atoms, not molecules, atoms, that have different numbers of neutrons, so one is heavier than the other. Don't confuse that with isomers. Isomers are molecules, not individual atoms, but entire molecules that have different arrangements or shapes. And a structural isomer is the example like they gave here, have a different overall shape. Geometric isomer, it's kind of like a structural isomer, except there's a different arrangement around a double bond. So the giveaway for a geometric isomer is usually that there's a carbon to carbon double bond. And what you notice in this example that they gave to you, you have carbon here, you have a carbon here, and then you have a CH3 here and a CH3 here. But on this one, you have them more diagonally oriented. Okay, fine. It just gives it a different shape, really. They come up with a new name for it, but really it's just a different shape. Now, the one that's kind of weird is called enantiomers. Enantiomers are molecules that look pretty much the same. They have the same chemical formula, but they are specifically, this one's specific, they are mirror images of each other. Like if I put a mirror right down the middle here, it could be like this molecule is looking in the mirror and it looks just like this one. Mirror images, like your left hand and your right hand are mirror images. But they're not the same. Like if you have you ever had like 2 gloves that were both the right hand and you try to put one of them on the other hand because you're too cheap to buy a new pair of gloves? It doesn't really work because then you end up with the hand pad part of the glove is on your back of your hand on one of them. So they're kind of the same, but they're mirror images. That's called enantiomers. Enantiomers are really cool because the thing about enantiomers is when you make drugs like pharmaceuticals, usually any drug you take actually has a mixture of 2 molecules. And they are enantiomers of each other. And usually one of them doesn't do anything. It doesn't hurt you, but it doesn't do anything. And only one of the enantiomers will actually be effective. So that's kind of interesting. All right. Dr. Baggett put this slide in, but I'm going to skip this cis-trans. This gets a little bit too much for me. Okay. So isomers are molecules that have the same atom components, but have different arrangements. You know, one molecule has a different arrangement than another one. That's really what I want you to know about isomers. Okay, functional groups. This thing you just have to memorize. I'm just going to be honest. You have to memorize this. These are little groups of molecules, groups of atoms that are commonly seen in larger molecules such that they have names as a group. And they give a certain function to the molecule. So the R in this picture is the rest of the molecule. It can be anything. But the functional group is what I'm circling. So these are little things that are attached to any other molecule. And when you see them, it gives the molecule certain characteristics. So what we're really talking about here is that when you think about molecules like water, it's simple because water has only two covalent bonds. They're both polar. Water's a polar molecule. But when you have large molecules, now we're getting into the level in the hierarchy of macromolecules. When you have this huge carbon chain, and there are different things attached on, not just hydrogen, but now these things, then the molecule, the macromolecule, takes on multiple behaviors, functions, based on what different things are attached to it. So the molecule is sort of a sum, has a behavior that's the sum of all of its little attachments. And these attachments we call the functional groups. All right. So each macromolecule contains certain functional groups. All right. And so in order to use the names of the functional groups, these are the names. And that you would know what I'm talking about. The only thing is you just memorize it. Hydroxyl is OH. Methyl is CH3. Carbonyl is how you pronounce that, is a carbon with a double bonded oxygen. And it's in the middle of a larger molecule. Carboxyl, now be careful because carboxyl is very close to carbonyl. It only differs actually by this one letter. This is an N and this is an X. So carbonyl is a carbon to oxygen double bond in the middle of a larger molecule. Carboxyl is a C, it's complicated, C double bond O, and then over here, OH. It's kind of like a carbonyl and a hydroxyl put together to make something new. So it's the carboxyl. The amino group is NHH, NH2. The phosphate group is P, 4 oxygens and 2 hydrogens. That's a complicated one, phosphate. And then sulfhydryl is SH. All right. The only one of these that doesn't take on any charge is the methyl group. The methyl group is hydrocarbon and it's nonpolar. You'll remember that if I draw it all out, CH3 would actually look, if we were really drawing it all out, it would be H, 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 oops. And then this would be the R over here. So remember that carbon to hydrogen, these are all nonpolar bonds. So the group itself is nonpolar. The rest of them are either polar or take on a full charge. So Carboxyl is a charged acidic. I'm underlining what I would really want you to know. Amino is charged and basic. Phosphate is charged and acidic. And sulfhydryl is polar. Now, how will you know these? If it were me, I would make this chart with just the names and then leave the rest blank and see if I could draw the structure and see if I could fill in what I underlined here from memory. Because again, you want to have it quick, hydroxyl OH. You don't want to be like flipping through or Googling it. If I say 8 times 4 and you have to Google it, it's just going to take too long. You got it, hydroxyl OH, it's polar. Methyl CH3, it's nonpolar. Carbonyl, C double bond O, polar. Carboxyl, usually we say COOH for carboxyl, even though it's C double bond O, OH for carboxyl, charged and acidic. Amino, NH2, charged and basic. Phosphate, H2PO4. How do you like that? It is charged and acidic, although a lot of times people ignore the hydrogens. I will tell you on the phosphate, the hydrogens, a lot, not that it's, you can do it either way, but you can just say PO4. The hydrogens usually release pretty quickly, so PO4 is phosphate. Either way, I don't care. And then sulfhydryl SH, polar. So this is one of those things you want to have pretty much at the tips of your fingers. So maybe flashcards could be helpful. Like I said, make a plain copy, make a plain chart like this and practice drawing it out and see if you can get them right quickly. And the reason I'm telling you this is that, you know, you're going to go into the next class, you know, and they're going to, on the first day, they're going to be talking about hydroxyl and amino and this. And if you never learned it the first time, you know, you may muddle your way through intro biology, but the next class, it's, you know, if you didn't learn it now, it's going to eventually, it's going to catch up to you. And I can't tell you how many genetics students you know, I'm talking about these things, you know, from the beginning when we start talking about molecular biology. And, you know, if you don't know your carboxyl, I mean, you're just behind. I mean, so now's the time to learn this and create this foundational knowledge, memorize this chart. Okay, so what do the functional groups do? Like I said, all the biomolecules that we're going to learn in chapter 3 start off with a carbon backbone, hydrogens, but then these functional groups attached on, and you're going to have to memorize in chapter 3 the more complex structures. So right now's the time to learn the functional groups so that when I start talking about bigger molecules, you know what I'm talking about. Okay, so there's 4, this is like a preview of chapter 3. There's 4 main types of macromolecules. The 4 categories are called carbohydrate, lipid, nucleic acid, and protein. And each of them is going to involve hydrocarbon plus certain functional groups. And we're going to practice identifying the molecules based on what functional groups you are going to memorize that they have. So anyway, that's a preview of what's coming next, but make sure you memorize this chart. I have some little practice sheets and such that if anyone's interested, email me and I'll send them to you and you can practice at home. And then there's, of course, you got your learning activities and your little practice quizzes and stuff that's already on eCampus. Okay, so that is the 3rd and final part of Chapter 2 lecture. You made it. You survived.